Hello students, I am going to take your geology practical class on cell biology in an easy way. My today's topic is cell viability study by Trypan Blue staining. I will discuss it in two phases. In the first phase, I will go for the materials to be used and how it will prepare it. In the second phase, I will go for the application and the result of the section. Okay, now let us go into the second phase. Now, first of all, I have talked about the first phase. The first phase, this is the material we will use. Now, in this test, a cell suspension is simply mixed with the dye and then visually examined to determine whether the cell take up or exclude dye. Then all the living cells are counted in percentage. So by this application of the dye, you have understood that it is a completely dye exclusion method. This method requires the following materials: Trypan Blue stain of 0.4 to 0.5% concentration, phosphate buffer of 1x strength, and a cell suspension as test sample. This is the main three criteria, and to react to it, for the reaction we should need the following instruments also. There is a micropipette of P 2000 Gilson micropipette, a fan drop tube in 5 to 6 in number, one centrifuge, one glass hemocytometer with 2 cover slip, and a syringe with 5 cc and 10 cc capacity, filter paper of number 1 Workman paper, or it may be 0.2 micron porosity, any type of filter paper, conical bottle 3 to 4, and a compound microscope also. Now with this structure, first of all, if we find the chemical nature of the stain, what will we see? We will see that this Trypan Blue has been formed from the toluidine. During preparation, 1 mole of toluidine is mixed with 2 mole of disulfonic acid. It is prepared in the alkaline medium, and thus the product has been formed a sulfonic acid of tetrasodium salt. This tetrasodium salt has carbon, hydrogen, nitrogen, sodium, oxygen, and sulfur molecules, having the molecular weight 960.8 gram. If you find the structure of this Trypan Blue, then you will find that here there is tetrasodium structure here, 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 and here, and also 2 azide chromophoric group, this one and this one. During this structure, by this structure it gains the different physicochemical characters. If you think about regarding the physicochemical characters, it is a vital stain. That means it has capability to stain the white living cells. It is an azo dye. Being an azo dye acid with a chromophoric group, has a strong affinity for cellulose, collagen, and amyloid structures to stain. Again, when it is a bluish gray in structure, then it is dry, but in aqueous medium, in solution condition, it looks deep blue in color. During formation in aqueous solution, the solubility rate is less than 0.1 milligram per ml. But commercially, you can get this structure, this stain, at 0.4% concentration. But this histological stain is carcinogenic in nature. That's why the washing with water and soap is must if you touch to skin accidentally. Now, I am going to prepare this 10 in the laboratory for your experiment. Okay. Now let us go into the preparation. Remember, it should be prepared fresh. It is prepared for quantitative analysis of morphological changes of the cell. We have to prepare it in two strengths, 10x and 2x. 10x is used for stock preparation of stock solution. For the 10x preparation, we should need the Trypan Blue, distilled water and sodium azide. In this method, the preparation method, 0.2 gram of Trypan Blue is taken in a conical flask. In this conical flask, distilled water of 99.8 ml is mixed with it. During mixture, the starter glass rod is stirred several times inside it until the mixing is properly made. After mixing, there should have some particles embedded in it. Maybe then it is required to filter. So second conical flask is taken and the filtrate is used, 0.2 micron filter paper. So any type of filter paper you can use, otherwise number one Whitman papers also may be used. Filtration is done to remove the sediments. Now after filtration, the filtrate is ready, but to stop any bacterial growth, sodium azide in 0.2% concentration is added to it, and then this mixture, this stain with 10x strength has been prepared now, and that should be used to prepare in our next stock solution, working solution, 2x strength. Now let us prepare 2x working solution. It requires some reagents like sodium chloride, distilled water and stock solution. In a conical flask, 0.42 gram sodium chloride is mixed with distilled water of 9.58 ml. This mixing is done by shaking the this mixture. Only 0.5 ml is of solution of saline solution is transferred into another conical flask, and in this saline solution of 5x strength, the previously made stock solution of 10x strength is taken into it. The proportion is 2 ml. So 2 ml stain is mixed with the 5x saline, and thus the stain of working solution becomes 2x strength. Now this Trypan Blue working solution will be reacted with the cell suspension of the sample in second phase to preserve it for the future. Now let us go to prepare the phosphate buffer saline. The phosphate buffer saline is prepared for the preservation and collection of the samples. During preparation, it requires sodium chloride, calcium chloride, disodium hydrogen phosphate, potassium dihydrogen phosphate. Along with it, you can also mix with it the magnesium chloride and calcium chloride. In different proportion, it is mixed in the aqueous medium to make it 100 ml solution. Thus, the reagent buffer solution 1x strength has been prepared. Now, why should we use them and how should I use them? We need it for the collection of the cell sample and as well as the preservation. After the preservation, for making the cell suspension, we also need it to mix with the cell sample. Other than these two, we also use it to wash the material which was used during the preparation of this procedure preparation, that is the materials. But the characteristics for which we have chosen this phosphate wafer as a definite chemical structure for its definite chemical features. It is isotonic in nature so that the cell structure remain intact. It is non-toxic in nature so that any cytosolic reaction do not happen. Being a both type of character, that is the monobasic and dibasic phosphate, and having the pH 7.4, it maintains the buffering capacity at wide range in the solution. The strength is 1x. The strength tallies with the strength of the working solution also. So thus we have prepared the phosphate buffer, and we have to keep it also for the use of the staining of the cells in second phase. In the second phase, we will make the cell suspension, stain, and count the cell with the help of these two prepared reagents. So my students, I have finished my class for phase 1 today. We will meet again in this phase 2 next time. Till then, thank you.